Thank you for being here today and welcome. That's what Dr. John Campbell always says. He goes, welcome as usual. Welcome as usual, or welcome back. A lot of what we're going to discuss now in chapter 15 is called the chromosomal basis of inheritance. Some of it will be review, but a lot of it will be an extension from what we talked about before, which is Mendelian genetics. So fairly straightforward dominance recessive kinds of inheritance. But there are lots of reasons we figured last time that there may be sexual reproduction as well as asexual reproduction. So in order to shuffle the genes and thereby ensuring there's a variety in our populations, which is better for natural selection. Give natural selection something to select from, different varieties. But there's other ways of getting variety. And so I'll show you what one of those is in a little bit. Yeah, so we talked about genes. They're located on our chromosomes. So here are pairs of nucleotides. So this might be G there and C there, and that might be A there and T there. G always pairs with C and A always pairs with T. And if we're going to be looking at this molecule on a molecular scale, we're going to be studying molecular genetics. And we do that in 1200. We get more into the detail of the DNA itself and how it confers traits. For now, though, we're looking at mostly population genetics. So how traits are inherited from one generation to the next. So the chromosome theory of inheritance states that Mendelian genes have specific loci and that chromosomes undergo segregation and independent assortment. Is that always the case? There are exceptions. Just like in ecology, there's an exception to everything. There's also an exception to these rules. So this is quite a long kind of complicated looking graph that you probably can't see very well. However, what it shows is yellow round seeds that are homozygous dominant crossed with green wrinkled seeds, which are homozygous recessive. And then you produce gametes, one chromosome each. We're looking at Mendelian genetics. And so what did we have in the F1 generation? Oh, well, we have both the dominant traits. So we have both smooth seeds, round seeds, and yellow seeds because they're dominant. And then we did another cross. And the law of segregation is shown with this cross because of the way the chromosomes are separated. So here in meiosis 1, the homologous chromosomes are separated in anaphase. And that's the law of segregation. In anaphase, the homologous chromosomes, along with their alternative genes, are segregated. Law of segregation. And are they independently assorted? Yeah. So in this case, we have a red homolog there, blue homolog there, blue homolog there, a different one, red one there. Well, we could get this combination, red and blue together. But we could also get these two ending up together as well. So that gives a bit more variety. And when they separate, then we have quite a specific combination. But there are variants of that combination. I didn't say that very well. However, the point being is that when the chromosomes line up like this, and say this is a different one. So they're lined up and they're going to go to either end of the cell. The side of the cell this one goes to is independent of the side of the cell this one goes to. So they're independent, the law of independent assortment. So we're working with fruit flies a lot in genetics because they have a 2-week generation time. They mate very, very quickly after they're born. They only have 4 pairs of chromosomes, so 8 altogether. And when we look at fruit flies, there's a term called wild type. That's the normal phenotype. And then alternative ones are called mutant phenotypes. It doesn't mean that necessarily that they're bad, although sometimes they don't function. So in this case, a red-eyed fruit fly is the wild type. They mostly have red eyes, but some of them, mutants, are white-eyed. And we use fruit flies. So we did a cross. We crossed a red-eyed female with a white-eyed male. And what you would expect in the F2 generation is a 3-to-1 ratio of red to white. So that's quite normal because the dominant allele will show even in the heterozygous individuals. So, but in this case, it didn't happen. Well, the ratio happened, but interestingly, only the males were white. So the white-eyed mutant allele must be located on the X chromosome because the female has the dominant wild-type allele that masks that particular trait of white eyes. So it's a mutant trait, recessive. So if the female has, all females are XX. If the female has one red eye allele, even if the other one has the white eye allele, she won't show it. But for the male who inherits XY, if the male gets the recessive allele on the X chromosome, the Y chromosome does not have the masking allele on it. They aren't homologous in that way. So it will be expressed. In this case, it will not be expressed because the dominant allele is there. So this was a discovery that transmission of the X chromosome in fruit flies correlates with inheritance of eye color trait. So that's some evidence that showed that a specific gene is associated with a specific chromosome, in this case, the X chromosome. And that's the case with humans. There is an Xlinked gene For color blindness, red, green. So if a female has it, but also has the normal one, then they'll just be normal color vision. But if the male inherits the recessive allele, there's nothing to mask that recessive allele. And it was 8% of males are colorblind. So here's some commonly studied organisms. Humans have 46 chromosomes. We often look at mice and mouse genetics. The fruit fly has 8, and this animal here, C. elegans, a little roundworm, has 12. But what's interesting about these individuals is that they share about 40% of the same genes as humans. So it's in these animals that genetically transferred diseases and that, like Alzheimer's and cancer and various things. That's how they're studied, their inheritance patterns. And there's lots of other animals that are studied as well. I think it's odd that a crayfish has 200 chromosomes. But the thing about the chromosomes that humans have, and very likely the crayfish as well, is a lot of them don't code for anything. So for humans, of the chromosomes that we have, and on those chromosomes we have 3 billion base pairs, or 6 billion bases, 6 billion nucleotides. I think it's only about 1% actually codes for anything. So a lot of it is non-coding, and it's thought that it's because some of those are left over from our evolutionary past. So we still have the DNA, but it doesn't get transcribed. So that was a case of sex-linked genes, or genes that are on either the X or Y chromosome. Linked genes, just plain linked genes, refers to genes that are on the same chromosomes. So sometimes, well, I mean, obviously loads of genes are on the same chromosome. So for the most part, these genes, like this one and this one, well, it'll always be inherited together because it's on the same chromosome. I won't say always because it's not always. Sometimes it won't, and I'll show you why in a moment. But here's a gene for erythropoietin, for example. That's a hormone for red blood cell production. And here's a gene for a cyst that causes cystic fibrosis on the same chromosome. So they could be inherited together. Each chromosome has hundreds or even thousands of genes. So these linked genes are located near each other on the same chromosome, and therefore tend to be inherited together, but not always. So this is a linked gene cross. In this case, it's a little bit complex because of the traits. They're not used to talking about these traits, but a wild type fly is gray colored with normal wings. So the little plus here indicates that that's the wild type. The mutant is black with vestigial wings or little small wings. So they did a test cross whereby they took a heterozygous individual and crossed it with a homozygous recessive individual. And you remember when we did that before with the flowers and the peas and everything, you would get a fairly specific ratio in a test cross. In this case, you didn't get what you expected. You got the wild type, so the parent. Yeah, well, you'd expect that with linked genes. You'd expect those both to be inherited together. And black vestigial, you would expect those to be together. But interestingly, you get these combos, gray vestigial and black normal, even though those are linked. Why is that? Why could that be? How could that be? So body color and wing shape are usually inherited together. So this shows the chromosomes, and these are the parents in the test cross. So this was the testing part, having the homozygous recessive individual. And that's the heterozygous individual for both traits. Yeah, most offspring you would expect would be a one parent or the other because those genes are linked. But where did the other ones come from? Where did those gray vestigial and black normal ones come from? Well, there is a way in which chromosomes are recombined. Genetic recombination is when the offspring, that's you and me, we have new combinations of traits inherited from the two parents. So of course we've got that. If we look at all of the genes that we've inherited from our parents, well, they're different. So we are recombinants. You and I are genetic recombinants because of the independent assortment of genes. Non-homologous genes. Well, there is another way to get a recombination, and that is crossing over of homologous genes or homologous chromosomes. So let's see what that looks like. So in Mendel's experiments, he observed, well, some offspring have combinations of traits that don't match either parent in the parent generation, and they're called recombinants. So a recombinant is anyone that shows new combinations of parental traits. And if 50% of all the offspring are recombinants, we say there's a 50% frequency of recombination. So let's look at linked genes. They're on the same chromosome, so you would expect them to be inherited together. If they're all independent, in a test cross, you would expect a 1:1:1:1 phenotypic ratio. If it's completely linked, you'd expect a 1:1 0:0 ratio. In other words, the offspring must be all parental. But there's an observation that about 17% of the flies were recombinants, in spite of the fact that their genes were linked. So a different frequency, less than 50%. Why is that? Well, that is because some chromosomes cross over. Yeah, they cross over. So during meiosis, the homologous chromosomes, there's one, And this is the homologue. They line up. And what happens is a phenomenon known as crossing over. So after they've crossed over, one of these homologues might have a little bit of the other one. So now you have a recombined chromosome. You started off like that with the same genes. So these two are sister chromatids. They're identical, so they have all the same genes. However, during this first phase in meiosis 1, there was some crossing over. So this one crossed with that one. And now you have a new combination. A recombinant. So we can use that to map genes. So we might know the genetic loci along chromosomes of lots of different genes. And we can, based on recombination frequency between the two genes, tell if they're close together or further apart. So in this case, for example, say we have, let's see, we have a gene here and here. Wait, I'm going to give that a different color. And there was crossing over. So now the recombinant, they have this one still, but now this one is on a different chromosome. That allele is on a different chromosome. It's not as likely to happen if they're close together. So in this case, let's see, there is one there and another one at that location. Well, that didn't cross over. So in the end, they'll still be on the same chromosome. So they won't be separated as often as the ones that are further apart. Stay with me. And then you can map where they are on the chromosome. So this is color. I believe that's color and something else. So these are two different genes. They're on the same chromosome. They're recombined about 9% of the time. The recombination of these two alleles is about 9.5%, and between these two, it's about 17%. So that corresponds to how far apart they are. Yeah, so CN and B are quite close together. CN and VG is also quite close together. But you get way more recombinations between B and vestigial because they're further apart. So they just tend to cross over more when they're further apart. And that's how genes are mapped on chromosomes. So I showed those chromosomes before, and they show exactly where certain traits are right on the chromosome itself. Now, you can't really tell absolutely, but you can tell relatively depending on how often this recombination occurred. Yeah, so this is a mapped a mapped chromosome. And it shows things like gray body, black body, where that is on the chromosome, different eye colors, wings, and other eye colors. Yeah. So these numbers here, they indicate the frequencies of recombination, and it gives you an idea where these genes might be on that chromosome. So sex-linked genes, X and Y, behave as homologs. Each egg receives an X from the mother. One sperm receives either an X or a Y. So there is a 50-50 chance of an offspring being male or female. So it's the SRY gene, or the sex determining region of the Y chromosome gene, that causes an embryo's gonads to develop into testes. If that gene is not present, then the gonads become ovaries. And the SRY gene, the SRY gene does regulate some other genes as well. But not all organisms are determined that way. Their sex is not determined that way. In fact, these are locusts, and they have an XO system, it's called. So females are XX, and the males are just simply X. There's no corresponding chromosome. In the ZW system, this is with chickens, ZW is female, ZZ is male. In the fruit fly, the females are diploid and the males are haploid. And with some organisms, like some reptiles, whether the offspring become male or female depends on how the temperature has changed for them. So if eggs are buried deeper in sand, for example, they might become males. And if they are not so deep and maybe cooler, they'll become females. So the sex chromosomes, though, they have lots of genes that are unrelated to sex or gender. But a gene that's located on either sex chromosome is called a sex-linked gene. And there's different patterns of inheritance. Some of them are disorders. So there's X-linked dominant disorders. And there are X-linked recessive ones. Color blindness is one. Duchenne muscular dystrophy is another. Hemophilia is another. So those are X-linked recessive alleles. And they're more often expressed by males. So an interesting thing that happens to females is called the Xinactivation in female mammals. Those are also known as bar bodies. So what happens is, as the female is developing, the cells are dividing. One of two X chromosomes in each cell is randomly inactivated. So that's during development. And then only one of the X chromosomes is active. The other one condenses and becomes a bar body. It condenses a lot like a chromosome. When it's assorted into the ova or the eggs, the bar body is activated again. But which X becomes a bar body is random in each cell. So that has implications. For example, this is a calico cat. And if a female is heterozygous for a particular gene, in this case it's fur color, it's located on the X chromosome, she'll be a mosaic. In other words, have both traits. Wherever that X chromosome is active, that's where that color will be. So in this case, we've got the orange color here. this X chromosome is active and the other one is inactive. As far as black fur color goes, this chromosome is active and this chromosome is inactive. And the odd thing is that you can see these bar bodies under a microscope because they're basically a condensed chromosome. And that's one way in which females are tested in the Olympics to see whether or not they actually are female. So apparently just in the old days, you know, you had to prove you were female if you were to join the Olympics as a female contestant. But I think they had to parade naked in front of the judges to prove they were female before someone discovered this bar body. So you take a swab of cheek cells and take a look at them under a microscope, and you can tell whether or not a person is female or not based on the presence of bar bodies. Well, that's not perfect, is it? Because you can be XX and have an SRY gene. You can be XY and not have the SRY gene. But also, you can have an excess of testosterone, for other reasons. And sometimes the adrenal glands are implicated in those reasons. So if there is an alternation of chromosome number, it can cause some genetic disorders. So during meiosis, which you noticed while you were studying it, the chromosomes are separated. But sometimes they don't get disconnected during that separation. So you have something called nondisjunction. And if it's a large scale, it can lead to spontaneous abortion or cause some development disorders. So nondisjunction, here we are in meiosis 1. And instead of the chromosomes neatly going to either end of the cell, these ones did not. They all went to one end of the cell during anaphase. And so you end up with these cells, once they've gone through meiosis 1, having 6 chromosomes. This one only has 2. And then you end up with gametes. In this case, they have 3, but the other ones just have 1 at the next division. Yeah, so that is nondisjunction in meiosis 1. You can also have nondisjunction in meiosis 2. So these are sister chromatids, and these ones separated nicely, but these ones did not. For whatever reason, it did not separate, and these chromosomes all went to one end of the cell. And then when they divided again after meiosis 2, this cell gets all 3, and this other cell just gets 1. And then this is normal. They get 2 each. And that's known as nondisjunction, when chromosomes don't separate into their holes of the cell. So it's called aneuploidy if offspring have an abnormal number of a particular chromosome. A trisomic zygote has 3 copies of a particular chromosome, and a monosomic zygote has only 1 copy of a particular chromosome. Polyploidy is another condition where there's more than 2 complete sets of chromosomes in an organism. So that occurs, it actually occurs quite a lot in plants. And sometimes their domestic plants are bred that way on purpose. It seems to make them more hardy. So Down syndrome is a result of an extra chromosome 21 called trisomy 21. So down here, there are 3 chromosomes instead of 2, nondisjunction. And so that occurs in a lot of different kinds of traits, some various development anomalies. Klinefelter syndrome is a result of extra chromosomes in a male producing XXY individuals. Turner's is a result of monosomy X, so producing an XO. karyotype. And yep, that is all I wanted to say today. One more thing. I'm just going to skip the next two. We're not going to talk about imprinting in this part of the course, but organelle genes are interesting because they are present in chloroplasts or mitochondria. So as we said before, if the mother's mitochondria are inherited by all of the offspring, yeah. So it depends solely on the maternal parent because the zygote cytoplasm all comes from the egg. Yeah, so in this case, there's an issue with chloroplasts. So the mitochondria have their own DNA, and chloroplasts in plants also do. And this harks back to the endosymbiont hypothesis that we'll talk about also a little bit later. So they have small plasmids, just small circular DNA. And in this case, there's an issue with the chloroplast a DNA that codes for chlorophyll. So some of these cells don't have chlorophyll. That's why they're yellow and they're not going through photosynthesis. Yeah, so I'd like to stop there. Thanks for watching.